Hi, my name is Kenton Chow, and today my group and I are going to be talking about the cat gene FeLD1 and why it should be modified. FeLD1 is a major cat allergen gene known as a secreted globular protein made from two heterodimers that each contain two polypeptide chains, chain 1 and 2, which are covalently linked by three disulfide bridges and are encoded by two separate genes to form a tetramer. It is speculated that its function is to protect the skin via homology with uteroglobin, which is used to protect mucosa or aid in the transport of lipid molecules such as steroids, hormones, or pheromones. Cats produce FeLD1 mainly in their sebaceous glands and is regulated by hormones, but can be found in large amounts in their saliva, anal glands, skin, and fur. Our goal is to genetically remove LD1 from the cat's genome using CRISPR-Cas9 technology in order to produce hypoallergenic cats. We've decided to target the FeLD1 gene because the domestic cat, also known as Felis domesticus, is known to be the number one major source of allergens within an indoor environment and the second major cause of involvement in allergic respiratory diseases. In general, cat allergies are one of the huge risk factors for rhinitis and asthma, which can develop into life-threatening situations. In Western countries, sensitization from cat allergies have increased from 10% to 30% of the general population. On its own, the FeLD1 gene causes immunoglobulin E antibody reactions in up to 95% of all cat allergenic subjects and accounts for 60 to 90% of overall allergenic activity in humans. Although there have been treatments to counteract the FeLD1 cat allergen, they mostly alleviate the allergic symptoms or have minimal success. For example, there's been development of anti-FeLD1A immunoglobulin Y antibodies that are put in cat food, which reduced the amounts of FeLD1 in cat saliva, which led to around 47% reduction of FeLD1 on cat fur. Another scientific group created a FeLD1 vaccine called Hypocat that causes around a 50% reduction in FeLD1 cat tear extracts. By incorporating CRISPR gene editing, the treatment won't just alleviate the symptoms of cat allergies, but remove the FeLD1 gene from cat cells and create FeLD1-free cats. The first step is to obtain DNA from cat cells where it will be extracted and purified in order to find the FeLD1 sequence from the CH2 heterodimer chain.
Hello, my name is Daniela Gonzalez, and I will be discussing the procedure in using CRISPR-Cas9 plasmid for our targeted sequence. In order to achieve hypoallergenic cats, the targeted gene will be removed by using the CRISPR-Cas9 plasmid, which will further implement the knockout of the gene responsible for FeLD1 protein production in vitro. To begin, we will first extract the cat's DNA from the cells and analyze the sequences that help produce FeLD1 protein. FeLD1 is composed of two polypeptide chains, which are encoded by two separated genes called CH1 and CH2. By disrupting the function of one of these genes, it will then cause the inability of FeLD1 protein production. Therefore, we decided to focus on the CH2 gene since we were able to locate the targeted genomic region for this particular gene. We will use about 20 nucleotides, which will be used as the guided RNA sequence. We will also have the polypeptide protein CH2 go through PCR amplification to obtain more copies of this region for further experimentation. After obtaining our targeted guide RNA, our next step is to search for a CRISPR-Cas9 plasmid. By performing gene deletion, it would ensure to block the cat cells from continuing to produce this protein since an indel mutation will take place from this cut site and which will cause the protein to fully shut off. The CRISPR that we will use contains the SB-Cas9 gene, which is an enhanced Cas9 gene with certain other components, and two sgRNA expression cassettes. Our guide RNA will be inserted in the first empty gRNA cloning site containing the U6 promoter, which helps achieve efficient editing. Expel restriction enzyme would also be added into the plasmid to make to ensure making the two cleaved sites of the DNA when inserted into the zygote. As a result, we will have a CRISPR plasmid with Cas9 gene programmed to target the sequence by using the guided RNA template. With these components, Cas9 will be able to sense and remove the targeted genome by cutting the double-stranded DNA containing the sequences responsible for the FeLD1 protein production. We would then proceed on to the next step, which would be to introduce the plasmid into a one-cell staged cat embryo.
Hi, my name is Jaylene Aguila, and I'll be explaining the process of taking an embryo and introducing it into a plasmid. In my research, I found an example on the website ncbi.gov where they extracted an embryo from mice, but the same process can be done to a cat. Some methods that can be used for producing animal models using fertilized embryos with the CRISPR system include microinjection, electroporation, and genome editing. But for the project, we will use microinjection, which is a method of extracting an egg to edit the gene by injecting the Cas9 and gRNA directly into the cytoplasm of the pronucleus of a fertilized embryo. The steps for the process of extracting an embryo is first, the synthesis and purification of RNA and DNA components. Second is the isolation of one-cell staged mouse embryo and microinjection of CRISPR-Cas9 components and transfer of injected embryos into pseudopregnant mice. And lastly is the genotyping of offspring to identify mutations.
Hello, my name is Christy Young-Tayag, and I will be discussing ethical considerations for our project. So ethical considerations if we were to genetically remove the cat's genome that contains the FeLD1 allergen gene in order to produce hypoallergenic cats. So considerations that we were thinking about was what would happen to humans and or the cats if we were to do this, and no effects will occur to the cat even though FeLD1 is naturally occurring in their genome. So no harm will come to them just because it is something that they naturally secrete and it's something that comes along with their way of life, and it doesn't affect how they live or any living processes that they have to go through in order to survive. It is just a trait that they possess to excrete the allergen. And if the gene were to be removed from the genome, it would just decrease the human allergies or eliminate or neutralize it altogether, as the cats will no longer secrete the FeLD1 allergen, or if they do, it will be at super low rates that will not be able to affect humans that have been affected by that allergy. Doing this through the genome will work on any cat, even if the cats have low rates of FeLD1 excretion. So it will probably just decrease the excretion rate even more or eliminate it altogether if the cat has a low rate of FeLD1 allergen. And since we're doing this using CRISPR technology, this technology has already been approved and researched. And so it has been approved for this process, and hypoallergenic pets already exist. So we would be able to utilize the process of using CRISPR-Cas9 in order to edit the genome safely, so no harm to the pets will come, nor to the humans, and we will just gain another friendly pet as a non-hypoallergenic one.
Hello, it's Daniela. For future works, we have considered ideas that can begin research in creating, like, for example, creating a serum that has the potential to use CRISPR-Cas9 plasmid to remove FeLD1 on existing cats instead of starting with an embryo. by producing the serum, it would at least be a bit affordable and increase the chances for more, especially long-haired cats, to be adopted. Hopefully, this can be further worked on to produce even hyperallergenic offsprings with just mating. Again, there was a study similar with food that maintained it, but it would have to be kept by the owner. So a serum or food would only be a temporary solution that could be fixed permanently by using CRISPR to alter the gene. So far, this concludes our presentation for on producing hypoallergenic cats.